All right. Welcome to Lesson 1.3 Scientific Method, Notes Part 2. So you're still going to use the same notes that you used for Part 1. You're just going to go down to the part where it says Notes Part 2, and that's what you're going to be focusing on. All right, let's do this. So, forming a theory. Theory is something that you'll hear thrown around a lot in common language, but it has a kind of different meaning in science. And it's really important to think about that meaning when we talk about something being a theory. So, a theory may be formed after the hypothesis has been tested many times and is supported by much evidence. So, our hypothesis, if you remember, is a prediction, what we think is going to be the answer to our research question. And we use that information that we've gathered to form a theory. Theories in science are based on lots and lots and lots of observation and evidence and testing. So, for something to be a theory, it has a lot of support. It's not just an educated guess. So, a theory is a broad and comprehensive statement of what is thought to be true. And a theory is supported by considerable evidence. So that evidence, and needs to go in your notes, that evidence includes observations, testing, data that's been collected over and over by many different scientists. All right, so let's practice using the scientific method. So here's our practice problem. You want to determine the effect of a certain fertilizer on the growth of orchids grown in a greenhouse. Materials that are available to you include a greenhouse, 100 orchid plants, water, fertilizer, and soil. You will want to know if the orchids will grow best with a weak concentration of fertilizer, a medium concentration of fertilizer, or a high concentration of fertilizer. So, how will you design an experiment to test different concentrations of this fertilizer? So, we want to know, does this fertilizer help my orchid plants grow? Right? So we need to make a hypothesis. So my hypothesis is my prediction or my answer to this question. Um, how does fertilizer concentration affect the growth of my orchids? So a possible answer. I predict that the orchids will grow best with a medium concentration of fertilizer. So remember, your hypothesis is your prediction or your answer to the research question. So, how do I set up a controlled experiment to test my prediction? So, this is a possibility for the research question in our example. So, if I have 100 plants, then I can divide them into 4 groups. So, my first group is going to be 25 plants that receive plain water, 25 plants that receive weak concentration of fertilizer, 25 plants that receive a medium concentration of fertilizer, and 25 plants that will receive a high concentration of fertilizer. The plants will be watered daily. Over a period of a month, the plants will be measured to see which ones grew the tallest. All right, so we kind of have to examine why I picked these 4 groups as the ones in my experiment. So first, let's think about controlled versus experimental. So what's my control group? You have to have a control group in order to see if your testing actually makes a difference. So my control group in this experiment would be the 25 plants that are receiving just plain water. And the experimental group would be the 75 plants that are receiving various concentrations of fertilizer. So it's a controlled experiment because all variables must be kept constant except the one variable that I'm changing. So what do I have to keep constant in this experiment. All plants have to receive the same amount of fluid each day. All plants should be grown in pots of equal size. All plants are grown at the same temperature. All plants receive the same amount of sunlight. We don't want the amount of fluid or the size of the pot or the air temperature or the amount of light to be things that affect how my plants grow, because then I won't know if my fertilizer makes any difference, right? So those are my constants or my controls: fluid, pot size, temperature, sunlight. And then what am I actually changing? The variable that's being changed is the amount of fertilizer received by each group of plants. So my control group is getting 0, no fertilizer. So that way I can see if fertilizer makes a difference. And also by having 3 levels of fertilizer, I can see how much fertilizer makes a difference. It's like a twofer. So after 1 month of measuring the orchids, the following data is obtained. So my control group, group 1, they grew an average of 15 centimeters. All right, that's about 6 inches. Group 2, that received a weak concentration of fertilizer, grew an average of 35 centimeters. That's over 12 inches, like closer to 18. Nice. Group 3, medium concentration of fertilizer, grew an average height of 28 centimeters. So that's a little under 12 inches. And group 4, which was my high concentration of fertilizer, grew an average of 10 centimeters. That's like 3 or 4 inches. So, looking at that data, is our hypothesis supported or not supported by our results? So remember, our hypothesis was that a medium concentration of fertilizer would result in the best growth for my orchids. Well, let's look at our data. Medium concentration of fertilizer grew 28 centimeters. That is not the highest growth. So that means our results did not support the idea that the medium concentration of fertilizer was best. So our results do not support our hypothesis. So our prediction was not supported by the observational evidence that we collected. So let's think about our conclusion based on these results. So we still learned something and we can still talk about what we learned. So our conclusion would be that orchids grow best with a weak concentration of fertilizer. So if we look at our data, 35 centimeters was the largest amount of growth. So at medium to high concentrations of fertilizer, plant growth is inhibited. Inhibited means that it's slowed down or blocked, especially those high concentrations. High concentration of fertilizer cause the plants to grow even less than no fertilizer at all. So some things you have to think about when you're designing an experiment. Why is it important to have a large sample size in any experiment? So it's important to test a large sample in order to get a true picture of the results of the experiment. If the sample size is too small, an inaccurate conclusion may be reached. Results obtained by testing a large number of individuals will be much more accurate than if only a few individuals have been tested. Instead of having 25 orchid plants per group, what if I'd only had 2 per group and 1 in each group happened to get sick? Now I have no real good support that what I did made a difference, right? Because 50%, 1 of my plants got sick. So individual variation happens, especially in living things. So the bigger number of subjects we have, the more those little variations don't make a big impact. Why is it important to repeat the experiment many times? So experiments should be repeated to see if the same results are obtained each time. This gives validity to the test results. Validity means that they are supported by evidence and repeatable. So we want to know that no matter who does the experiment, we're going to get the same results. That's really important in science. What's the importance of the control? The control shows what will happen when the experimental factor is omitted or not there. Without the control, there's no basis for comparison, and you would not know how the experimental factor affected the results. Think about our plants, right? The greatest amount of fertilizer caused a growth rate of only 10 centimeters compared to our water group, which was 15 centimeters. We might not have known just how negative an impact high concentrations of fertilizer would be if we didn't have the control group to compare it to. So, how is a theory different from a hypothesis? And they are very different, and it's important that we understand this. So, a hypothesis is an educated guess, although I hate the word guess, but this is how most people explain it. It's testable through observations and experimentation. And normally it's not just, I guess randomly. I've already made some observations. I've already asked some questions based on a phenomenon I've seen. And that information is used to make my hypothesis. So it's not a just out of the blue guess, it's based on things I already know. A theory is a broad statement of what is believed to be true based on many experiments and considerable amounts of data. Why is it so important that a scientist accurately describes the procedure used in an experiment? Well, by accurately describing your procedure, it allows other scientists to repeat the experiment and verify the results. Remember, we want validity, meaning that we can say that anyone who does this experiment is going to find the same results. And that's really important. Also, if your results are something like this procedure or this drug helps treat cancer, we want to make sure that other people can follow those same procedures to have that same success. What is the difference between an independent and a dependent variable in an experiment? So this should just be a review. Remember, my independent variable is the thing that I change. Independent, I change. So it's the thing that is deliberately changed by the scientist. The dependent variable is the one observed during the experiment. Dependent variable is the data that we collect. So dependent variable is the data that depends on the independent variable, the thing I change. In a controlled experiment, why must all the variables except one be kept constant throughout the experiment? If several variables were changed at the same time, the scientists would not know which variable was responsible for the observed results. Was it the fertilizer, or was it the amount of sunlight, or was it the amount of water? We're not going to know if we change too many things at once.